substances entering and leaving plants. The cell membrane controls the movement of substances into and out of cells, as shown here. The cell membrane therefore controls the chemical composition of the internal environment of the cell. The cell membrane controls movement in two ways: passive processes, in which the cell does not use energy, and active processes which need energy. Passive processes. These work on the principle of diffusion, in which molecules and ions in solution move from areas of high concentration to areas of low concentration. In the situation shown, diffusion will result in the sugar molecules being equally distributed in the water. Cell membranes are selectively or partially permeable. This means that some molecules and ions can pass freely through them down concentration gradients, but others cannot. Oxygen, carbon dioxide, and water are examples of molecules which cross membranes easily by diffusion. In a respiring muscle cell, for example, oxygen will pass into the muscle and carbon dioxide will pass out. Osmosis. The cell membrane is partially permeable in that it allows some molecules to diffuse through rapidly, but slows down the passage of others. The glucose molecules try to diffuse to the outside, but cannot get through the membrane because the pores are too small. Water molecules diffuse through the pores down a concentration gradient. This membrane-controlled diffusion of water molecules is called osmosis. Osmosis can work both ways, so that water can pass into or out of the cell. An experiment to demonstrate osmosis.
So if we look at this, you notice that that tubing put into pure water. So water will enter the tubing and will cause the tubing to swell. Look at this other experiment here that they are demonstrating. The container is full with sugar solution, and the tubule or the tubing here, which is a tubing like plastic, so things can move in and out. Now look what had happened to it. Okay, let's return to the first experiment again. Just look at this. Remember, the water here is pure water, and this tube contains sugar solution. Okay, remember we talk about osmosis as the movement of water from an area of high concentration to an area of low concentration. So what we have here is that this water here would have been your high concentration. Inside here, it is a whole lot of sugar, very little water. So it's a tendency of the water, therefore, to move from where there's pure water and try to get into the bag there with that solution. And we see what happened there. As we look, we see that increases in size. Okay, look at the second experiment. It's the opposite of what happened. Over here, let me clean up this. Okay, over, come on. Oh, so over here, we have the sugar now. So that is full with sugar. And over here, we have, this is pure water. So you expect the opposite to occur. The water will move from this and go. So we have a movement going all the way from that water until you reach a, so let's look at, see what's happening here. Let's undo. Okay, let's see what happened there. Oh, take my thing off, right? No. Stop. Here we go. So we see what's happening there. Good. So that explains what happened from a visual level, what we are able to see. Okay.
In this experiment, fresh potato was diced, surface dried, and weighed in 2 separate batches. The batches were then treated in the following way. The first batch, A, was placed in pure water. The second batch, B, was placed in sugar solution. After 2 hours, the batches were surface dried and reweighed. Calculate the percentage weight change and place the figure in the table.
Okay, so this is a simple operation for you. Now we're looking at the percentage weight gain or weight change. So this one was put into pure water, similar amount of potato, and this one was put into a sugar solution. Now, as you see here, the beginning there, this was 15, and this was 16, and we notice that this now become 15 point what? In B, which is this one, it become 15.2. So right away we see that this one what? Right, decreases, and this one was 15, and it's almost 16 now. So we see an increase here. So 15 over 15.9 over 15 times 100, and you do your calculation there. Then, so this is what happens.
When the cell becomes limp because it loses water osmotically, it is described as flaccid. When the cell becomes rigid because water has been taken inosmotically, it is described as turgid. Eventually, the vacuole shrinks so much that the cytoplasm is pulled away from the cell wall. The cell is plasmolyzed.
So 3 terms here that I want you to be familiar with. One, here that we use and we say the word flaccid. All right, it is limp, soft, and this is when the cell loses water. This one is turgid, means that the cell gain water, as in this case, it becomes firm because the cell wall is pushing back against the contents of the cell in the vacuole. And then this one here, the term plasmolyzed, we're saying that this has passed flaccidity. It is reached to a stage that at this stage, it's unlikely that the cell will be able to come back from that stage that it is in now, okay? So the 3 terms that you must be familiar with: flaccidity, turgidity, and plasmolyzed. Flaccid cell is when the cell loses water. Turgidity is the result of the cell gaining water. Look at animal cell now. Okay, in an animal cell, So we have an animal cell here. It is placed in a dilute solution, meaning that you have more water than sugar, basically. And if we're thinking about lemonade, this does not taste good. It tastes like water, not enough sugar. So the water enter by osmosis and the cell will now burst. Remember that this cell doesn't have a cell wall. So once it expand, it will burst. In this case, it's placed in a concentrated. So this is a very sweet, syrupy kind of sugar and water. And then after a while here, this is what will happen to the cell. The cell will become shrunken. Okay, so we see some difference here between the plant cell or the animal cell. In a plant cell, remember we use the same word, turgidity. It will become turgid. But in this case, it becomes turgid, but if it goes further, it will rupture. In this one, it will shrunken. And then in this case here, we talk about plasmolyze in the plant cell, but this one we will talk about here. It will be crenated, or what we say crinkled or wrinkled, okay?
Active processes.
Okay.
It is possible to track.
Now, so this is where we're going to stop today. We're not going to move to the active one today. So we look at passive, which is just diffusion, in which the water move, or the substance, not water necessary, substance move from a high concentration to a low concentration. Osmosis is the movement of water. So when we talk about osmosis, student, remember, we're talking about water. So just keep that in mind. Now, there are some terms that we are using here that you must know. When we talk about a turgid cell, what happened? You must be able to describe that cell. The cell is going to get more round and firm in a plant cell. So we say it's turgid. And if it's an animal cell, it will eventually rupture the cell. If it's a plant cell, it's put in a concentrated solution, it will cause the cell to lose water. The plant cell will become flaccid and eventually plasmolyze, but the animal cell, we say it will become wrinkled or crenated. So that's it for today. I'll be sending you some worksheets so you can practice, watch this video over and over again, and make sure that you get the point that we are talking about.